This is how prokaryotic replication forks work. The process by which some prokaryotic cells like E. coli replicate themselves is called binary fission. This is a form of asexual reproduction that allows one cell to divide and become two new daughter cells. The genetic information in the two daughter cells is completely identical to the one parent cell. This replication of genetic information called DNA happens before cell division. The DNA of most bacteria is contained in a single circular molecule called the bacterial chromosome. The origin of replication denotes the area of active replication called the replication fork. DNA replication will begin at a specific area of the molecule called the origin of replication. Primase is an enzyme that synthesizes short RNA sequences called primers. These primers serve as a starting point for DNA synthesis. Helicase attaches to the replication fork and moves into the fork, breaking hydrogen bonds as the replication fork moves along the DNA. DNA ligase in E. coli, as well as most prokaryotes, utilizes energy gained by cleaving NAD to create the phosphodiester bond, binding single strands of DNA at the base pair level. Single-stranded binding proteins stabilize the unwound DNA. Gyrase is a topoisomerase that relieves supercoiling of the double-stranded regions ahead of the replication fork. Supercoiling occurs when DNA coils back on itself when it is overwound or underwound. Replication of both strands happens at the same time. The two strands of DNA are antiparallel, meaning they run 5' to 3' and 3' to 5'. DNA replication happens in the 5' to 3' direction. Because of this, there is a leading strand and a lagging strand. Okazaki fragments are caused by the discontinuous replication along the lagging strand. For the segments on the lagging strand called Okazaki fragments, there are 1,000 to 2,000 nucleotides per section in prokaryotes. DNA polymerase I serves to connect Okazaki fragments by deleting RNA primers and replacing the strand with DNA. DNA polymerase III is essential for the replication of the leading and the lagging strands, whereas DNA polymerase I is essential for removing of the RNA primers from the fragments and replacing it with the required nucleotides. These are my citations, and for creativity, all illustrations